Hello students, today our topic is MHC, that is Major Histocompatibility Complex. It is a set of surface proteins, or we can say glycoproteins, which are located on the cell membrane of nucleated cells. So the fragment molecules of MHC are generated inside the cell when the antigen is digested by the antigen presenting cells, and these MHC molecules act as a cell surface vessel in order to display the antigen molecule onto the T cell receptors. So the name MHC got its name from the fact that the genes in this region encodes the proteins, and they determine whether the tissue is being transplanted between the two genes, the two individuals will be accepted or rejected. So it is the main function of MHC is to identify, so if it is going to recognize or it is responsible for the genes which are playing main role in the acceptance or rejection of graph. Therefore, the main role of MHC is to identify the antigen between self and non-self body. So, intercellular, it will recognize the antigen as self and non-self. Then, intracellular recognition will occur, and ultimately it represents the antigen to the surface of antigen presenting on the surface of antigen presenting cell. Histo refers to tissue, and the compatibility refers to living harmoniously together. So MHC molecules are responsible for always recognize T lymphocytes. There are two types of MHC molecules which worked in the immunity, that is class 1 and class 2. Although there are four types of MHC molecule, but the main role is placed by only class 1 and class 2 MHC molecule, because class 1 MHC molecule recognize TC cells, CD8 positive marker of TC cells, and class 2 MHC molecule recognize CD4 positive molecule of helper T cells. So if we define MHC, it is called as the complex is trans membrane attached protein which work on recognition of antigen between self and non-self, and also responsible for antigen presentation. Now, these MHC molecules are found in in the blood sample of mice, and it was first recognized by Peter Gorer in 1930, and they identified and designated MHC as four groups of different molecules. Then in 1980, George Sennel, Jane Dossett and Barry received the Nobel Prize for their contribution in the discovery of MHC molecule. So there are MHC genes which are responsible for the genes which are present on the 6th chromosome of humans and 17th chromosome of mice, which are responsible for the formation of class 1, class 2 and class 3 MHC genes. So class 1 MHC genes are encoded and expressed on the surface of nucleated cells, all the nucleated cells, and they are recognized by TC cells, that is CD8 positive marker. And class 2 MHC genes are expressed on antigen presenting cells, and they are exogenously recognized by CD4 positive molecules of TH cell. And class 3 MHC molecule encode for different immune function. They are basically involved in the formation of complement system, and they are involved in inflammation. Class 4 MHC molecule are found in only leukemia cells. So first is class 1 MHC molecule. Class 1 MHC molecule is the group of first group which their molecular weight is is 45 kilodalton, and they are as I told you, on the surface of all nucleated cells except nervous tissue and platelets. They are recognized by cytotoxic T cells and responsible for CD8 positive marker, and it brings cell mediated immune response. If we see the structure of class 1 MHC molecule, there is alpha 1 chain, two domains basically, alpha and beta. Alpha domain comprises of alpha 1, alpha 2 and alpha 3 molecule, and there are approximately 90 amino acids which enter into the transmembrane domain into the into the cell, antigen presenting cell. And beta domain is a single domain which forms beta strand, and it is present between alpha domain and the transmembrane domain of the class 1 MHC molecule. So the binding of the structure, there is a deep groove in the there is a deep groove between alpha 1 and alpha 2 molecule of alpha domain, and there are 8 parallel chains in the beta domain, and this alpha domain is present on the top of the surface of class 1 MHC molecule, and there are 8 to 10 amino acids which are responsible for the formation of this deep groove inside the alpha 1 and alpha 2 domain. And the three strands, the beta plated sheets are being organized along with the alpha domain, and these beta sheets form anti-parallel strands of amino acid, and this alpha 3 domain is highly conserved in nature, and this structure of class 1 MHC molecule belongs to is similar as that of Ig molecule, and they do not express on the cell membrane of tumor cells. And next, the main function is the antigen is internalized, processed by the proteasome, and peptides are produced, peptides which are further loaded into the groove from the endoplasmic reticulum. And in this groove, peptide groove, the antigen is going to bind. Next is class 2 MHC molecule. Class 2 MHC molecule, there are two different domains. One is alpha domain and beta domain. There are two alpha 1 and alpha 2 domain, alpha 1 and alpha 2 chain in the alpha domain, and beta 1 and beta 2 in the beta domain. Alpha domain is of 33 kilodaltons and beta domain is of 28 kilodaltons. Now the difference between alpha 1 and alpha 2, as you know that there were 3 alpha domains in class 1 MHC molecule, but in this there are 2 alpha domains and 2 beta domains, and both the domains form a transmembrane complex, and the peptide groove is deeper as compared to the class 1 MHC molecule. This is you all know that it binds, that it recognizes CD4 marker of TSL, and it binds with the exogenous antigen only, and antigen is processed inside the endosome, and the peptide groove is then present on the antigen presenting cell. And in class 3 MHC molecule is involved in complement system cascade, and it is involved in inflammation, and the main components complements include C2 molecule of the complement system, C4 molecule, and factor B. Class 4 molecules are not, the complete structure and mechanism is not studied till now. They are basically found present in the T cells of leukemia and some immature thymocytes. Then is beta MHC molecule is also named as HLA. In humans, it is found in the 6th chromosome of leukocytes, and it is responsible for Mhc molecules in the Mhc genes in the human beings. HLA is the set of surface protein present on the all nucleated cells. So basically, as I told you in the starting, that Mhc molecule are responsible for the differentiation of antigen into self and non-self. Therefore, it is if the Mhc molecule are going to recognize self molecules and antigen, it will mount the autoimmune response. So, along with Th cells and autoantibody production, the basically Mhc molecule are responsible for this. Graft is rejected by Mhc genes because Mhc genes recognize the graft as self or non-self. Adaptive immunity, they are responsible for adaptive immunity and defense against infection. Sometimes they are expressed in cancer cell destruction also. And Mhc is the general term refers for the cell surface antigen of vertebrates. If we talk about mouse, H2 complex is known as Mhc genes in mouse. It is the cluster of genes responsible for the production of antigen located on the nucleated cells and complement components. Also, the H2 genes are present on the short arm of chromosome 17 in mouse, and it consists of structural genes. The genes that make up histocompatibility complex are known as haplotypes. The major function of Mhc molecule is degradation of proteins, Mhc molecules are loaded with the sample of peptide fragment from the degradation of protein. As I told you in the starting, that they are glycoproteins, and this peptide is the mirror image of proteins present inside the cell. So Mhc molecule, the major function is to recognize the antigen as self as well as non-self. If it is going to recognize the antigen as self, then it is going to mount a damaging effect of immune response, and if it is going to recognize that as non-self, then the immune response is elucidated or immune response is exhibited, and they bring out defense against infections and diseases. As I told you, that if Mhc molecule are going to recognize self molecules as non-self, then they are responsible for the creation of autoimmune diseases, and every person has a peculiar spell of them. This is due to Mhc molecules. Summary, that you all know that class 1 and class 2 Mhc molecule are responsible for antigen presentation, recognition of self and non-self. They are encoded by the chromosome 6 of humans, and there are two types of antigen proteins which are degraded by the Mhc molecule and are involved in the neutralization of antigen. This is all about Mhc molecules. Thank you.